An antigen is any substance that triggers an immune response, including allergens that trigger allergy responses and pathogens, infectious, non-self antigens that cause disease. Pathogens include prions, viruses, bacteria, fungi, and parasites. Bacteria or viruses are pathogens that can cause infections such as a common cold, sore throat, or tinea, whereas protists are the disease-causing agent in malaria, whilst prions cause diseases like mad cow disease. The first line of defense. Organisms have a range of physical, chemical, and microbiological barriers to keep out pathogens. Animals have skin that acts as a physical barrier between invaders and your body. It's a waterproof mechanical barrier that pathogens can't get through unless it's broken. The human respiratory and reproductive tracts are lined with cells that secrete mucus, which traps invaders, and then cilia that line the respiratory tract and pushes mucus up the throat where it can be coughed out. Tears also help to prevent pathogens from settling on the surface of the eyes. Chemical barriers in animals include mucus, which also contains lysosomes, enzymes that break down the cells of certain bacteria, and the highly acidic environment of the stomach, which kills many pathogens that could enter the body via food or drink. And microbiologically, we have the microbiome, which is beneficial bacteria that grows on the skin, in the bowel, and other places in the body that stop other harmful bacteria from taking over. However, sometimes pathogens breach the first line of defense. Cells within the organisms have MHC1 markers to distinguish between self and non-self antigens, and therefore, if a non-self antigen is detected, the cells can initiate an immune response to remove it from the organism. When the immune system detects a pathogen, it responds in a variety of ways. If pathogens manage to pass through the first line of defense, a series of cells are engaged to fight the pathogen. Phagocytes are the first immune cells to deal with invasions of a pathogen. Neutrophils are a type of phagocyte and are the most abundant phagocytes, and they're the first to arrive to deal with invading pathogens. Once they've engulfed the pathogen, they self-destruct. Dendritic cells are phagocytes that have an important role in connecting the innate and adaptive immune system by presenting antigens of the foreign material to activate T cells and are known as antigen-presenting cells. Macrophages are also antigen-presenting cells and are phagocytes which can engulf and digest pathogens continuously and digest the foreign material by extending long, finger-like extensions and pulling them into the cell body. Natural killer cells detect foreign material and secrete death ligands to induce apoptosis in the infected cell, destroying the cell and infected agents. Apoptosis is a controlled process of cell suicide. It occurs in response to a specific cell and its nucleus and involves an orderly series of biochemical events. The cell and its nucleus shrink and there is an orderly dissection of chromatin. Death is finalized by a rapid engulfment of the cell, dying by phagocytosis. This safely disposes the remains of the cell. The complement system. The complement proteins circulate the blood and assist the body's defense mechanisms. Once the proteins encounter a foreign body, they become activated, which results in enhanced inflammation and phagocytosis of pathogens and destruction of cell membranes. Opsonization is when proteins attach to invading microorganisms so that they are more readily identifiable as foreign to the phagocytes. There is also the membrane attack complex, which destroys membranes of invading microorganisms by creating pores in pathogens known as the membrane attack complex, causing the organisms to explode. This is known as necrosis. Once selected, a healthy T cell inflammatory response. The inflammatory response can be caused by invading pathogens, burns, chemical irritation, trauma, or tissue damage. The inflammatory response attracts phagocytes and promotes healing. It involves 3 stages. The first stage is the increased diameter and permeability of blood vessels. So mast cells release histamines that cause blood vessels to vasodilate and permeability to change. This increases blood flow to the area and allows defensive substances to leak into tissue spaces. The second stage is phagocyte migration and phagocytosis. Within 1 hour of injury, phagocytes appear in the scene. They squeeze between cells or blood vessel walls to reach the damaged area where they destroy invading microbes. The third stage is tissue repair. Since the functioning cells or supporting connective cells create new tissue to replace dead or damaged cells. Some tissue regenerates easily while others do not. The third line of defense, the lymphatic system. The lymphatic system is a system of lymphoid organs, including the thymus, bone marrow, spleen, and lymph nodes, and lymph fluid that are involved in transporting lymphocytes and removing foreign matter. A colourless fluid leaks out of the circulatory system into the tissues. This fluid is called lymph, and some of it is absorbed by lymph vessels, where it is then transported back to the heart and deposited back into the bloodstream. On the way to the heart, it passes through areas of spongy tissue called lymph nodes, the secondary lymphoid tissue. It is in the lymph nodes where lymphocytes, specifically B and T cells, are found, as well as phagocytes. If, for example, you get a cut and your skin breaks, allowing pathogens to enter into your living tissues, an antigen-presenting cell which has MHC2 markers on it, for example, a macrophage, will then transport the pathogen to the lymph node. Inside the lymph node, there are helper T cells, which all have unique T cell receptors on their surface. An antigen-presenting cell presents the antigens from the pathogen it has engulfed and starts to bump into helper T cells. Eventually, it'll bump into a T cell with a matching receptor to the antigen, which will then stimulate it to replicate, making thousands of copies of itself. Each of these replica T cells have the same T cell receptor as the parent cell. Therefore, there are now thousands of T cells with the complementary receptor to the antigen being presented. And when this happens, we say the B cell has been selected. Once selected, a helper T cell comes that also has a T cell receptor that matches the antigen, binds to it, and secretes a cytokine called interleukin. Interleukin causes the B cell to make clones of itself, which is paracrine signaling. Some of these cells are B memory cells, and they remain fairly inactive, living for many years, giving you lasting immunity against that pathogen. The other clones are plasma cells, which produce antibodies at a very fast rate and release them into the lymph, where they flow into the circulatory system. This means that antibodies spread across the body and can attach to antigens in the body, forming antigen-antibody complexes. This means that the antigen cannot affect the body or cells, so they are out of action. Hence, the antibodies contain them until a phagocyte can come and engulf them. This also activates the complement system and is referred to as the humoral response. Cytotoxic T cells are involved in the cell-mediated response. They are also found in the lymph nodes, and when an antigen-presenting cell presents an antigen with the matching T cell receptor, they will bind. A helper T cell will then come along, secreting interleukins and initiating for the cytotoxic T cell to start replicating and making clones of itself. A cytotoxic T cell will come across an infected cell, for example, a cell infected with a virus that has MHC1 markers on its surface, as well as an antibody that matches that T cell receptor. The cytotoxic T cell responds by releasing a death ligand, stimulating that cell to undergo apoptosis.